Structure and function of synapses. What is a synapse? What functions do synapses perform? So a synapse is where an axon of a neuron connects with the dendrite of another, or connects with an effector, which can be a muscle or gland. So they link neurons together and coordinate activities. Impulses from one neuron to another is by a chemical which is called a neurotransmitter. And there is a small gap called a synaptic cleft, which is 20 to 30 nanometers wide. The neuron that releases the neurotransmitter is called the presynaptic neuron, and the end of this axon ends in a synaptic knob. This contains mitochondria and endoplasmic reticulum to make the neurotransmitters. And the neurotransmitters are then stored in synaptic vesicles. Once the neurotransmitter is released from the vesicles, it diffuses across to the postsynaptic neuron, which has receptors on the membrane. So looking at the structure of a synapse, this is the axon or nerve fibre. The end of that ends in a swelling called the synaptic knob. There's mitochondria, vesicles containing acetylcholine. Presynaptic membrane is this part, and then this is where the acetylcholine is released into the cleft in response to an impulse reaching the synaptic knob. At the other end, on the postsynaptic membrane, there are receptors for acetylcholine if it's a cholinergic receptor. And then there's also acetylcholinesterase, which is an enzyme which breaks down any excess acetylcholine which is left over in the synaptic cleft. So the functions of synapses, they transmit impulses from one neuron to another, and they act as a junction, allowing a single impulse to be transmitted to many different neurons at a synapse. It allows a single impulse to stimulate many responses, or a number of impulses can be combined at a synapse, and this allows stimuli from different receptors to interact, a bit like how the rod cells converge to one bipolar neuron. So some facts. The chemical or neurotransmitter is made only in the presynaptic neuron. Neurotransmitters are stored in the synaptic vesicles and released into the synapse when an action potential reaches the synaptic knob. The neurotransmitter diffuses across the synapse to receptor molecules on the postsynaptic neuron, and the neurotransmitter binds with the receptor and sets up a new action potential in the postsynaptic neuron. Excitatory and inhibitory neurotransmitters. So excitatory neurotransmitters depolarize the postsynaptic membrane, making it fire an action potential if the threshold is reached. Acetylcholine is an excitatory neurotransmitter. It binds to cholinergic receptors to cause an action potential in the postsynaptic membrane. Inhibitory neurotransmitters hyperpolarize, preventing an action potential from firing. GABA is an inhibitory neurotransmitter, and when it binds to receptors, it causes potassium ion channels to open on the postsynaptic membrane, hyperpolarizing the neuron. Synapses become fatigued. When many action potentials arrive in a short time, the cell runs out of neurotransmitter. The impulse can't cross the synapse. This is known as adaptation and means animals ignore stimuli that last a long time, like the tick of a clock, because it's irrelevant to their survival. Features of synapses. They are unidirectional. So synapses can only pass impulses in one direction, from the presynaptic neuron to the postsynaptic neuron. Summation. So low frequency action potentials don't produce enough neurotransmitter to trigger a new action potential in the postsynaptic neuron. Summation involves building up neurotransmitter in the synapse. And there are two types. There is spatial summation. This is seen with the rod cells which converge to one bipolar neuron. So it's where a number of different presynaptic neurons together release enough neurotransmitter to exceed the threshold value of the postsynaptic neuron and therefore trigger an action potential. Temporal summation is where a single presynaptic neuron releases neurotransmitters many times in a short period. So it increases the frequency. If the total amount of neurotransmitter exceeds the threshold of postsynaptic neuron, then an action potential is triggered. So here's some diagrams of spatial summation and temporal summation. So this shows that each individual neuron on its own doesn't release enough neurotransmitter, but when they both release neurotransmitters, that is enough to fire an action potential. This one for temporal summation, if there's a low frequency of action potentials, only a small amount of neurotransmitter is released, which is below threshold. It's the high frequency of action potentials, it releases lots of neurotransmitter above the threshold, and therefore an action potential is triggered in the postsynaptic neuron. And there are inhibitory synapses as well as excitatory ones. So inhibitory ones, on the postsynaptic membrane of some synapses, the protein channels for chlorine can be made to open. This leads to inward movement of chlorine, making the inside of the postsynaptic membrane even more negative. This is known as hyperpolarization, and it makes it less likely that a new action potential will be created. These synapses are called inhibitory synapses.